Hello, everyone, to the second instance of the AIRR-C seminar of this year, 2025. I'm very excited to have two speakers today, Steve Bosinger from um, Emory University in the US, and then also Maria Francesca Abate from École Normale Supérieure as our junior speaker today. I will just briefly introduce Steve before his talk, which will be on a broad survey and functional analysis of Ig loci variation in Rhesus macaque. So Steve has received his PhD in microbiology and immunology from the University of Western Ontario. While there, he focuses research on studying the pathogenic events in early HIV and SAV infection. And he completed his postdoc work at the University of Pennsylvania. It was there where he began his work studying African monkey species, especially a specific species which avoids AIDS despite lifelong SIV infection. He was awarded a number of fellowships and Young Investigator Awards, and today his lab main research focuses on the viral pathogenesis of SIV and SARS-CoV-2 in the primate model and how B cells are programmed after vaccination. And I'm very much looking forward to your talk, Steve. Thank you.
Thank you. I think I need to update my intro because I'm not so young investigator anymore.
Yeah.
Um, but well, thank you for inviting me to this group. As an immunologist, it's fun for me to come into a new field of what I think are mostly geneticists. So it's nice to make new friends and new colleagues. I'm going to talk today about a project that we've been working on for several years with probably groups that are a little bit more familiar to those of you in this community, particularly Corey Watson. And really, the genesis was that of this project was to try and use genetics to better utilize the monkey model, particularly the Indian origin Rhesus macaque model for HIV vaccination studies. So to give some background on this, why would the genetics of this notoriously complex loci and that's very hard to study be relevant for HIV vaccines? And that is largely because the current best candidates we have for HIV vaccines are very different from those for conventional vaccines. And that is with most vaccines, you don't really particularly pay much interest or have a goal to try and target uh, target germline immunoglobulin alleles. You're usually just using an immunogen to try and get an antibody response against a defined structural target, and and whether the response is polyclonal or monoclonal, as long as you get neutralizing activity, you're good. With HIV, those attempts largely were failing to even get neutralizing antibodies. And so the field as a whole, about 20 years ago, decided to um, go from a ground up and invest a lot in basic research to try and understand what an antibody that could neutralize the diverse, strains of HIV that you'd see would look like. And so a protocol was in place by multiple groups to look in people in sub-Saharan Africa and people in the United States that were chronically infected with HIV and look at these few people that were able to provide that had serum neutralizing antibodies and what we called and people that had the 5 or 10% of these people that with their serum you could neutralize over 70% of the available strains at high potency. And so this coined a term in the field that we call broadly neutralizing antibodies. And so then essentially in a nutshell, what the field has been trying to do for the last 15 years is reverse engineer where these antibodies came from in terms of their unmutated common ancestors, and then try to engineer vaccines that will engage these particular alleles and nurture them along the way to become what we hope are mature broadly neutralizing antibodies. And so this is referred to as the germline targeting strategy. And there's several reviews that will go over this. And after many years in this field, in 2022, the first paper showing that a germline targeting allele was capable of eliciting antibodies of the right genotype was published by Bill Schief and his colleagues, and furthermore, last year there was the first proof of principle showing that a germline targeting immunogen in non-human primates was capable of eliciting the proper alleles. And so this was really a big shift for the field, not only because of this success, but also in thinking about how we do immunology and how we do vaccinology for HIV vaccines. And so for 20 years, our metrics of success for these vaccines has been the eliciting of some kind of neutralizing antibody. But, beyond the sort of landmark results of being able to elicit these antibodies, the secondary thing that come out of these papers was that these vaccine trials are now really relying on a genetic readout to see if they're on the right track or not. Basically meaning even if you're not getting neutralizing activity, showing that your vaccine is able to elicit the proper class of antibodies is going to be critical for continuing to engage that immunogen with more trials and more studies. So where does that leave us for genetics in the monkeys? Well, first of all, some of the other things that come out of these studies were that depending on which allele you had would really depend on whether or not in the humans, whether or not you could make a neutralizing response. And this is for the VRC01 class antibody. And second, your genetic makeup of that particular allele would determine your functional usage in the naive repertoire. And so when it comes to monkeys, we have a few problems. One of them was that for some of these alleles, particularly the VRC01 class allele that is the leading candidate, the VRC01 class antibody for which the VH1-2 allele is the leading candidate in the most advanced in clinical trials, we haven't to date found a close homologue of that. Second, knowing that some of these alleles are going to depend on having genetic makeup, we didn't really have a good characterization of a lot of the macaques in our centers to know if they were going to have the proper allele or not. So when starting out to do this project, that was addressing some of those goals were what we had in mind. And as I start out, I want to point out that there's been a lot of work that has been done in this field by a number of groups. The IMGT has done work to characterize a monkey that was sequenced by Pac Biotechnology and assembled, which is the MM-Mule10 assembly. Ramesh and Kim Sorelli with Shane Crotty's group have also done work to try and build an assessment of and at least characterize the alleles that are available in monkeys, and I'm putting some of the statistics here. And lastly, I really want to point out the contribution of Gunilla Carlson-Hedestam and Martin Corcoran, who have also really spent a long time working at characterizing macaque alleles to be able to address these issues and have a very popularly used database called KimDB that we use and a lot of have used. However, when we were moving forward, we wanted to be able to try and build something that was going to be relatively comprehensive for the field, and that was going to incorporate not just repertoire sequencing, but also we wanted to be able to incorporate DNA sequencing to make a stable genotype of all these animals. Trying to get a nomenclature was going to be difficult because we didn't understand haplotype diversity. And lastly, there was still a lot of a dearth or a missing of data for the light chains that we want to address. So what are some of the challenges if you were trying to take on this endeavor and build a comprehensive resource of monkey immunoglobulin alleles? One of you can approach this technically on two different ways. One is by sequencing the genome, typically with long-read sequencing. This is challenging, of course, because of the complex structure of the IG loci. There's lots of duplications and pseudogenes. There are a lot of structural variants, and so you'll have, what looks like a particular allele or gene in one animal that might be in a completely different area in another. And it's also still a challenge to understand from sequence alone which of these alleles is actually functional and used in the actual B cell repertoire. So the alternative would be using repertoire sequencing, which has the benefit of identifying sequences that are in the functional. Can you hear me?
Bonjour Vincent. Je te dérange pas là? Non, 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 non. On parlait de toi justement,
All right.
avec le Chili.
En bien ou mal?
On bien parce qu'il m'a demandé.
Thank you. All right. So repertoire sequencing is a very highly popular a way, an effective way, and convenient way to address this. However, there are other caveats to that, and one is that, of course, you're going to have in your repertoire, even if you target what you hope are naive sequences, you're going to have a fair amount of somatic hypermutation, which necessitates doing inference. You're going to not understand the genetic framework and where these particular putative alleles are going to be lining up structurally in the DNA. And then also things that are at a lower frequency may be missed. And you might miss some sequence data in some of your approaches. So before I get into our technical approach, just to try and understand what is the basis of providing monkeys to the HIV enterprise in the United States, I wanted to try and show the major suppliers of monkeys. And so we have what's called the National Primate Research Center Network. And so these are 7 different primate centers and an 8th one in Puerto Rico that are funded, or subsidized heavily by the NIH. And these have been most of these have been established in the 40s and provide probably the vast majority of animals for these types of studies. This is further supported by a group of not nationally funded, but private foundations, private research centers that are providing monkeys as well. All of these centers, this is just if I were to paint a broad stroke, have, let's say, in excess, roughly 1,000 to 2,000 Indian origin Rhesus macaques. And another sort of minor caveat to this is that in the United States, at least, we typically will try to recruit only Indian origin, not Chinese origin Rhesus macaques, and that will have some of the implications on our study while we're focusing only on Indian. All right, so I've talked about the technical challenges trying to get at a resource or high confidence resource with these very challenging genetic and RNA sequences. And so in our initial plan that we put in for our grant to do this work, we proposed that we were going to do long-read targeted long-read sequencing of captured immunoglobulin sequences. And this was where we took all known immunoglobulin sequences from monkeys to date, built a substrate, and this was done in the laboratory of Corey Watson and Melissa Smith, built a substrate to enrich the immunoglobulin DNA, subject it to long-read PacBio sequencing, and assemble the IG loci and then annotate. On the other hand, we were going to be also taking a separate sample from the same monkey for all these monkeys and doing heavy and both light chain repertoire sequencing, inferring the germline, and then using this data to work with each other to get a highly valid, or what we hoped was going to be a highly corroborated set of sequences that we then posted publicly. So this was the original plan. This is what actually happened in real life. We had a large number of collaborators that I want to point out as I get into this to do all this work. First, my collaborator on this, Corey Watson, Melissa Smith, and Swati Saha, have done the PacBio side of the DNA sequencing that's shown up here, including designing the oligo substrates, doing PacBio sequencing, doing assemblies, and then also using a separate set of whole genome sequencing to try and build a set of references as our starting data point. My lab with Stacy Lapp and Amit Upajay, who's a bioinformatician, have been handling the RNA sequencing side of things. And so we've been doing AIRR-C and recruiting a lot of the monkeys from the centers around the United States. Once we generated the DNA data, we needed to have a reference set we need to be able to pull the alleles from the DNA. And so we collaborated with William Lees, who had made the Digger algorithm, which was able to analyze the sequences and get putative IG alleles, and also built us a reference set. And lastly, Gur Yari, Ayla Perez, and Viraj Pine from Bar-Ilan and now Yale University who did a lot of the very heavy lifting of the bioinformatics to try and get a highly confident set of alleles inferred from this data. So this was essentially what went into this, and I'll go into more details, but this is what, at the end of all this work, we have generated what we call the Macaque United Set of Alleles, or the NUSA, where we have not just the sequences of the allele itself, but also surrounding recombination signal sequences, leader sequences. We are working to make unique permanent identifiers and then maintaining identifiers across the various databases that I've shown. And then we also have references back to the individual animal and the data sets that each sequence comes from. Okay, so to make that more simple, when we got our grant, our initial objectives was to conduct high-quality haplotype reference assemblies, and I'm not going to cover that today. That's the work by Sooty and Corey that I'm going to put a teaser for potential future AIRR-C talk. And our, what I'm going to focus on today was constructing an accurate IG gene catalog across all these primate centers I've talked about. And the data set that I'm going to be presenting today, I need to note up front, is highly biased towards animals that were from my center at Emory. And this is largely because these were the ones we first got samples and first got sequenced into the pipeline. So any of the sort of estimates of variation are going to be, taken with this sort of balance in mind. Okay. So step one was that to be able to annotate the DNA, we needed a set of base reference of Rhesus IG alleles that Digger could use as to try and understand the similarity. And so for this, we pulled from all the existing data sets that were published by various other groups, which gave us our what we call our base set. In this, we had over 1,000 V alleles and over 500, heavy chain V alleles and 500 and 300, respectively, kappa and light chain alleles. And these are the references that we built this from. We also used some novel data that we had generated in-house from some of the whole genome sequencing and assemblies that Corey and Sooty had been generating. All the DNA was sequenced and assembled using the HiFi and then annotated using the Digger tool. And then for the repertoire sequencing, we annotated the sequences based against the Digger information, and then any alleles that we couldn't find evidence for in our base set or in the DNA, we then inferred using the Tigger tool and the Piglet tool. And this was able to give us a set of germline alleles from the RNA repertoire data. Okay, so once we had that, we were able to come up with a set of alleles that we saw in our cohort of animals where we had both DNA and RNA evidence. And so this is the big, exciting table that we came up with from all that Herculean effort of sequencing, where we had in our data set characterized with both DNA and RNA evidence, 700 heavy chain V genes, 500 and 300 light chains, respectively. If we were to compare that to our base reference to ask, okay, out of all of these genes or alleles that we've characterized, which are shown in yellow here, so these are alleles where we saw them, both DNA and RNA, at least once, usually more times in our animals. How did that overlap with the existing set that we started from? And you can see that in most cases, about a third were novel. If we break this down into numbers, our germline catalog extended the known alleles for heavy chain V genes by 26%, 49 for kappa V genes, and 47 for the lambda V gene segments. And so all of this data compiled together, we call the Macaque unified set of alleles. If we asked what was the distribution of these alleles, you can see that there was a lot more. What I'm showing here is the allele counts if we look at just the genomic DNA alleles that were pulled by the Digger algorithm compared to ones where we saw both in genomic and RNA. And you can see, as you'd expect, that there's a lot, this is a more restricted data set, so there are fewer alleles here. And we do have, it's kind of an interesting biological question. Why are there so many alleles that look real? They have RSSs, they match up to known sequences, but we can't find them in the repertoire, despite sequencing over 100 animals. And so Down here are the sort of reference statistics. So these are the number of alleles that we're finding on a per animal basis as a median for these three loci. And so it kind of matches up with what you'd expect. Um, we also asked how many of our alleles are seen in other databases. And so this upset plot summarizes information where we were seeing about 473 of the heavy chains that were novel and only seen in ours, and most, and over half of those were seen in both the both the DNA and RNA, with about 40% in just the RNA. Okay. Next, we wanted to try and get the beginning of a nomenclature system going for these, not a final nomenclature system. And so to do this, in lieu of having structural data available to try and give these more firm names, we used the allele similarity clustering system that had been developed by Eilert Perez and Guriari's lab, and essentially this is acting as a proxy for assigning these to actual IG genes, and instead putting them into families based on sequence similarity. And so, because we don't have the second criteria here to comply with IUIS nomenclature, and so essentially, to boil it down, gene family is having greater than 75% nucleotide similarity, and to be contained into an allele similarity cluster, it has to have greater than 95% similarity. When we looked at the structure of our data with this, we had, I think, I'm going to get the number wrong, I think it was approximately 68 families for the heavy chain genes. Each one of these nodes here is an ASC, or an allele sequence cluster. The ones that are colored means that we saw them in our cohort with both DNA and RNA. The ones that are shown in gray here, we didn't have both DNA and RNA evidence for. And so, as we were seeing, there's a large family of what we call silent alleles that we're seeing in the DNA but not in the RNA yet. If we break it down into families, there were a large number of ASCs that were containing, a single member, but the majority were containing, somewhere up to 30. The highest, most frequent allele had 128 separate alleles inside of it. In terms of subgroups, we saw some expanded subgroups in the realm of about 25 groups within the set of ASCs contained with itself. Having all the sequence data also allowed us to look at the RSS sequences, so the recombination. These are sequences, of course, that are used for the recombination of naive B cells, of developing B cells as they develop into mature naive B cells. And so, what we found, typically at the 5' region, you're going to have your septamer and your nonomer at the 3' region. And what we found was that in general, between the heavy chain Vs and the kappa V genes, there was a lot of repeats of similar sequences and usage of a similar sequence. We found two strong consensus sequences in the heavy chain V genes. But what was interesting was that any sequence here that's noted with an asterisk is seen in other loci. So what was interesting about the lambda was that we saw the most frequent RSS sequences actually seemed to be unique to this loci, not seen in the other ones. The other thing that was unique was that there were a few sequences, and these are noted by red here, that we saw that were not seen in human, and so they might be unique to Rhesus. And so we're not sure what the significance of that is, but it was just interesting to note that you can get this from the data, and some of these were actually quite frequently in the top 3 or 4 used consensus sequences. We also asked, what is the sharing? So how common are these alleles between individuals in our database? And you can see it, as you might predict, there are actually a few common alleles that were present in, over 90%, sorry, not 90%, 90 individual animals out of our 106 subjects. And then there were a fair amount that were similarly unique. But you did have a high degree of allele sharing in general. And so, the majority of our data set was not simple singletons. It was, in fact, the opposite. Most alleles were seen in multiple animals. The same was essentially true for the ASCs, and that we saw a high degree of these allele similarity clusters across individuals. Very rarely did we see ones that were just in a single animal. The other question we wanted to know was, okay, so you have all of these novel alleles. How different are they from what's already out there? And so in this analysis, we simply computed the number of nucleotide substitutions and amino acid substitutions that were different from the closest allele in our base set. So in the starting data set we started with that contained all the previously published alleles. How different were they? As you might predict, that a lot of the vast majority were off by a single SNP. However, we did have many that were had more than that. We had many that were up to 5 or 10 nucleotide changes. Okay, so now we're going to get into what would be utility. We wanted to be able to make this database, but how can we employ this? What use does it have? And so one of the first analyses that we did was to try and apply this to single-cell RNA-seq data to see if it was going to improve our estimate of somatic hypermutation rates. And so, many of you probably do either 10x or plate-based single-cell RNA-seq. You will generate BCR sequences from these and then want to understand potentially how close that is to germline. And the standard essentially has been to make an individualized germline for each of the animals in your study, and that would be by usually using an inference program like IGDiscover, and that will give you probably the closest estimate. And what we found was that essentially as you would predict, the individualized database had the most modest number of mutations. And if we compared the number of sequence differences we were seeing in our recovered data when we used either IMGT, an individualized database, KimDB, or our new MUSA, and what we're computing here is the difference. We are seeing that most of the time, in using IMGT as a starting reference, you actually had very few. You had only close to 60% that actually had an exact match. In suggesting how close the starting database was. This was vastly improved by using KimDB, where compared to the individualized, you were seeing almost 97% agreement between using KimDB as a starting database. And then using the MUSA as a starting database was slightly improved, about 1%, where we saw that, out of this, we had 19 sequences out of our 1,800 that we were detecting something different or something different away from the individualized database. Where the MUSA really, I think, makes a difference is with the light chains, and that is largely because there's still not a lot of light chain information out there. When we compare to IMGT, we were seeing that in most cases for kappa, there were almost 40% of the starting sequences were not similar, were not identical to that in IMGT, whereas we had 92 exact matching sequences, or so 92% of our sequences were exact matching, and this goes up to 97% as composed to only 76. So we feel that the information here will help for the light chain estimates of somatic hypermutation greatly. All right. Lastly, one of the interesting things is, okay, when we've generated this large data set, can we ask now how close are these monkey or these Indian origin rhesus macaque IG alleles to known human V genes that are important or biomedically important for these broadly neutralizing antibodies? And so I'm just showing a subset of the data here. We have a larger table that is going to be posted online very shortly. And what we're finding is that in a lot of cases, we were finding actually, substantial increase for some of these BNAbs, where, what looks like I'll use the term homolog, not ortholog, but what looks like a very more close homolog between rhesus and human does exist with this additional sequencing. The other thing I want to point out, however, is that there's been a long held belief in the field that the IGHV1-2 allele does not have a close homolog or ortholog in the Rhesus macaque. And this was a previous estimate by, a very limited data set done by Ramesh and Thomas Kepler and Danny Dewick. And when we've expanded this analysis, at least to the animals in our MUSA, which is heavily biased to the Emory Primate Center, we haven't seen a great improvement. We've seen, about 1%, but we haven't yet found something that's above 95% that would say, oh, this allele is out there. However, for some of these alleles, we are seeing improvements. And so we feel that as we expand more of our sequencing into additional centers, we're likely going to find closer homologs. And so this is relevant because, as I stated, in the beginning of my talk, we have new data now that is showing that if you put germline targeting immunogens into macaques, you can get engagement of these alleles. So understanding the constitution of our primate center colonies and understanding which of these alleles have these, which of these monkeys have the allele that's necessary is very likely going to be more and more incorporated into HIV vaccine studies. And the last thing I want to point out in regards to is in regard to the VH1-2 allele, which is the allele that's used for the VRC01 broadly neutralizing antibody, is that separate to our studies in a different group of monkeys, people have been successful in vaccinating these animals and getting what they term as VRC01 class antibodies, and these are based off the virus target that the antibody is targeting, which is the CD4 binding site, but not the genetic sequence similarity between these targeting antibodies and the VH1-2. And so this is more empirical evidence that if you are getting a germline targeting SOCIP, it's not going to be targeting the VH1-2. That something like that would be in humans. Okay. So where do you find the data? Thankfully we collaborate with William Lees and Guryari, who have done an amazing job of trying to make these data sets not only publicly available, but available in a way that's usable and searchable and dynamic. And so this MUSA data set has now been posted on VDJBase as a reference book. Gur and Eilert are maintaining a GitHub where the entire data set can be downloaded as a CSV file and filtered or manipulated, and sequence extracted based on the user's needs. And lastly, we have a preprint out now that we are hopefully getting published soon that people can see the entirety of the study. So to wrap up, so I've got 5 more minutes. The MUSA, expands this catalog of immunoglobulin germline genes. And what's important to note is that we've done this where we're focusing on alleles where we have both DNA and RNA evidence. Where we have discovered a number of alleles that are closer to the human genes for some of these biomedically important V alleles or V genes, sorry, and increasing our sequencing to centers beyond what we've done currently is likely to find more of these. We feel that the MUSA greatly improves the availability of light chains for doing somatic hypermutation analyses. This set of alleles has revealed some orthologs that are closer than was much previously appreciated. We're not there yet, but we want to get to the foundation for a genetically guided allocation of the Rhesus IG loci, where we can start to be able to recruit animals into studies with their IG alleles known beforehand rather than doing it after the fact. And then lastly, to try and get towards a unifying annotation, the MUSA, combined with structural genetics we feel is going to provide the foundation for a nomenclature that'll be stable. So what's next for Rhesus IGNX? And this is just my last teaser slide. I just want to point out, the work by Swati Saha and Corey Watson that have started with a trio of animals taken from the Emory Primate Center that they've used to benchmark all of the capture data that they've done, and have also done a series of other trios from other primate centers to try and get a handle on the very extensive haplotype diversity. And so that is something that's really underlaid our data that I talked about today, but I didn't really get into it, and then hopefully they can have a chance to present that at a later seminar. And so the most important slide is who did the work. And so I really want to highlight Ahmed Upajay, and he's not named here because I took his acknowledgement slide, and so he didn't name himself. But Ahmed Upajay in my group has really been leading the informatics on my side, and Stacy Lapp has really been leading all of the RNA sequencing events. Uh, Adam Erickson, our calling geneticist, has helped recruit the appropriate animals, and then our sequencing facility has done a fantastic job. Um, a lot of the bioinformatics heavy lifting here was really done by Gur's lab, particularly Ayelet and Vered. And so I really want to just point out their efforts. And then, this work would not be possible without sort of the collaboration from Corey and Swati and Melissa, who have sort of really, I feel been leaders in the IG genetics field. And lastly, I want to point out, work by Ganilla to get this field really going and help us in our initial days. And finally, you need a partner as well to advocate for your science. And so John Warren, our program officer, has really been advocating for monkey genetics to be, put into a more central role with the HIV. So that's my talk, and I'd be happy to answer any questions.
Great. Thanks so much, Steve. That was fantastic and really a lot of work. I'm sure there are many questions. I'm just gonna start. So you said in your summary slide that the more animals you would look at, the more alleles you would find. Do you think there is an upper limit, or will we just find more and more?
I think, that's been something in the field where, we had thought initially there might be an upper limit from some of the earlier inference studies, doing uh, attrition is the wrong word, but those, the curves where you add
Yeah.
up the additional animals. Um, but having this data set and seeing, having this data set, seeing the number of new alleles, which, and then also seeing, Corey and Swati's data where there's remarkable haplotype diversity across animals and also within an individual animal. We only have for that data set, we only have two centers, and we're seeing a remarkable amount of data or sorry, of differences between those the haplotypes and at the Oregon Primate Center and the Emory Primate Center. So I think once we start getting a handle on the data from the other centers, I think there's going to be a lot of new alleles. I think that's my opinion, but I do think there's going to be a lot. Um, we're not, we have pulled back on the number of animals we're sequencing. Before we were trying to get about 100%, and now we're going to be going down to 50 because I think we will get the information there. But yeah, that'll be a very interesting. Once we start having, the data I showed was one center, but largely one center, but once we have 6 or 7 in there, I think that's going to be interesting. Maybe then we'll start hitting the asymptote.
Great. Felix?
Well, we're kind of following up on that. I had two questions, but I think you said your animals were mostly, I can't remember, Indian macaques or something.
Yes.
But so do some centers focus on other, I don't know if the word is subspecies or other geographical groups or.
Yeah. Well, to not bore you with a history lesson, the centers were largely founded off Indian origin macaques, and then India stopped the export of macaques back in the late 70s. And so then some other centers, well, not other centers, but there were research efforts in Chinese macaques. Why, and so you'll find in European primate centers you'll have more of an even balance, and there'll be more of an influence, more work done on Chinese. The original viruses that became what we now call the SIV Max were found in Rhesus by chance in the US primate centers. They'd actually come from African species, gotten into the Indian origin macaques, and then were cloned and isolated, and those became the founding viruses that we used. And so there was sort of this momentum that when if you took those viruses and you put it into Chinese, it was different enough that the pathogenesis was lower. So this became the high bar to use. Now it's all different because when we're doing a challenge, we're actually using SHIV, so a SHIV, what we call in our field, where we have basically a human envelope put onto an SIV. And so we really haven't tested SHIVs in Indian versus Chinese, but because, there've been 20, 25 years of using Indians for this for that reason, the field is largely stuck with that. So a lot of the primate centers in America they've really invested in making sure that they bred their Indian origin rhesus and maintain those. But in Europe, it'll be different. There's more of a balance of Chinese and Indian because of these historical reasons.
I mean, just from understanding total diversity, I mean, you'd also want to do these other types, obviously, I guess.
Yes, absolutely. And what I kind of glossed over here was in Ganilla's KimDB paper, she did include Indian and Chinese in there. I was only restricting what I showed in ours to the Indian. Um, in that paper, I think it's about 330, something like that. It's over 300 alleles that she described as Chinese only. And when we've looked in we have this expanded set of animals now, and we've seen that I'm going to get wrong with the numbers here, but about I'd say it's over 100. We've seen about 100 alleles are actually found in the Indians. So I think this will be, these data sets as we evolve, we'll be able to figure out what there are. But there is going to be diversity, that's for sure. About a decade ago, primate centers were slightly concerned about potentially having monkeys that were intergressed, like where the baby was, Indian and Chinese parents, and so had done a lot of work to sequence genomes and see that there was, they are different. And so that's why we've kept these colonies sort of pure.
So there's no macaque mulata from Africa?
No, no, they're Asian.
Oh, okay. Sorry.
Yeah, yeah, macaque,
Um,
yeah, yeah. No, yeah,
okay.
that's the again, I don't want to go down the history lesson,
Yeah,
but
yeah, yeah, that's okay. Yeah, yeah.
no. But what's important to understand is the SIV. If you go to Africa and you look in African monkeys, there's over 40 different SIVs characterized, each from its own African species in the wild. If you go to macaques in India and China, in Asia, you will never find an SIV. It's not naturally found in them. It was the SIVs that became used for 10, 15 years actually just came from macaques being housed in the United States with African animals, and that their virus is getting into the macaques. So,
Okay.
yeah,
Yeah. I was also wondering, you have all those alleles that don't get expressed, and then you showed all this variability in your RSSs, and I guess is it possible that there's just a huge number of non-functional RSSs that we don't know about, or
yeah, that's one of I think the fun things we can do with the data that GUR is planning on doing, where we can actually, because we have both, we can actually say, is there, we haven't done that for this first paper. We're just trying to get all this data out there. But yeah, trying to see if there is sort of sequences that are more heavily utilized in the express repertoire versus not, I think will be, helpful for, potentially building better ways to annotate genomic DNA.
great, thank you.
Any other questions? Then I have a few more questions.
Sure.
So you mentioned this huge diversity, and which will probably grow as we look at more individuals and so on. But my immediate kind of desire is to know, what are they here for? What are they doing? How can we get closer to actually understand what all of this diversity is about? And how does it make the repertoire different in terms of also binding landscape between the individuals and so on? Yeah.
Where do I start to answer that? I think there's depending on how granular you want to get, having an ability to attach repertoire to antigen binding specificities or antigen binding in general. There are technologies such as LibreSeq where you can either sort antigen-specific B cells that are specific against a particular epitope or particular antigen. You can build tools where you've barcoded the epitope called LibreSeq. So we've done some of those studies, and what we've seen is if you are looking, if the question, if I'm going to rephrase the question twice, but I'll do it once. One way, if you're saying what is the diversity in the antigen-specific repertoire, at least with we've done a number of studies with one particular immunogen, known as BG505 SOSIP, which is an HIV envelope that's been heavily utilized. And in some cases, we get neutralizing activity, and we've used single cell sequencing to characterize the BCRs in the antigen-specific B cells with LibreSeq. And whether the animal was neutralizing or not, we've actually seen a fairly oligoclonal set of alleles. So, if there's a neutralizing antibody that's responsible for 90% of the neutralizing activity of that animal, you know, I'd say something like 60-70% of the alleles that we find in B cells that are binding to that antigen are from that heavy chain or a slight variant of that heavy chain. So it does tend to be very oligoclonal. Like you'll get sort of at least one or two dominant clones, and then a couple smaller ones, and then singlets. But it is, which was surprising, to see that it's that sort of dominated by just a few clones.
But then
Um,
why do we need so many different versions of it across individuals? So how does that arise? If within one individual it's mostly oligoclonal, if it's enough, so to speak, to make a good neutralizing response.
right. So if you have, I mean, I'm not an evolutionary biologist. I would say, I'll just say diversity is good. No,
Group
you know,
selection. Yeah.
yeah. I mean, yeah, maybe it's group selection. I mean, yeah,
Corey has a strong response to that. Yeah.
yeah. I'm sure, I'm sure he does. Yeah.
He
And
says
then
no.
what? He says no. Okay.
Yeah, I mean that's that's one of the question that is of very strong interest to me, right? We have all these alleles, but what are they there for? I mean, it's nice to have all these catalogs, but yeah, it would be nice to understand why nature came up with this catalog, kind of. Um,
Well, hope that's one of the. It's not the primary goal of our
oh, I know, I know.
very utilitarian use that I focused on maybe. But one of the other things, I'm not saying it's not an important question. It's on the evolutionary side, hopefully having the repertoire data and the frequency data will help us to understand it a little better. And maybe it is. I mean, again, I'm gonna say something that's probably gonna make all the geneticists go crazy, but if we do see that there are particular RSSs that are slightly divergent and those sequences aren't in the used repertoire, do we feel that those are just vestigial sequences?
Mm,
You know? Yeah.
mm, interesting. Very hot take. Um, could you go to the slide again where you show the difference in SHM statistics if one did it in different ways?
Yeah.
Yeah. Can you explain again the x-axis? Difference in SHM frequency, but what kind of frequency? Is it nucleotide?
Yeah.
Yeah.
So these are what we were comparing was, oh, and I didn't update it to MUSA. This is a previous name, but we settled on MUSA.
Mm.
Um, what we were saying was essentially if you used, we had generated for a number of animals, I think it was 15 animals, single cell RNA-seq data sets.
Mhm.
Then, from antigen-specific B cells. And then we compared those so once we got the BCRs from those animals, we compared that to a database. And so what we used as a reference was we had also generated IgM libraries, either IgM or IgD. We had generated IgM or IgD libraries from these animals and done IG Discover to make inferred individualized databases.
Mm.
And then we compared how many sequence differences we were seeing when we compared one of these query databases against when we took that data and said, okay, what's the somatic hypermutation rate? And then what is that, how does that increase compared to the individualized? And so we were seeing that you saw a large increase when you compared IMGT because it's lacking a lot of these alleles, right? But less so with these. Does that answer your question? Yeah.
Yes, yes, yes. And did you also look into, whether clonal families are more correct or like are changing in size or things like that, and whether how trees look different when you use, the wrong reference and the more correct reference and things like that? So not only focus on the particular.
We haven't done that. No, we haven't done that. I think we plan on publishing this paper, and I think someone will want to benchmark these things, and I don't know if it'd be fair if it's us,
Oh.
So,
unbiased benchmarking, I like.
Yeah.
Yeah. Okay. Do we have any more questions from the audience? I think no. So thanks, Steve, again. This was really, really nice. And
Thanks for inviting me.
thank you. Thank you.